before we start talking about the new parts, how did people do in terms of just finishing the kind of little discussions for any, yeah, are there any, someone want to wrap up? What was kind of the take-home messages from part 1 of our chemistry review lab?
Buffers resist change in pH.
Yeah, so Stephen was saying buffers resist change in pH. And what was your kind of empirical evidence that helped you see that?
Well, the observations that we did in the experiment that Dr. Tina did and with you, that in water, when adding the solution, the acid and the base, the changes were first very sharp and then gradual, and that didn't happen in the buffer solution.
So exactly. So like in your discussion, you definitely need to bring in your actual data, your actual observations. Like you can't just say, oh, I saw buffers resisted, you know, I see buffers resist change in pH. You definitely want to be very explicit about, you know, this is how I know. I saw this and this is convincing me of this. So the next parts of the lab, part 2 and 3, are going to be looking at, you know, electrolytes, things that are ionically bonded like sodium chloride and potassium chloride and calcium chloride, and how they react in terms of dissolving in water or nonpolar solvents like oil. And then also looking at the same kind of behaviors, but with a carbohydrate, which in particular, we're going to look at glucose. What kind of bonds hold glucose together?
Covalent.
Yeah, covalent. Those are, and is it polar or non-polar covalent?
Polar.
Polar.
Polar. Again, with sugar, you've got like that oxygen in the ring, which gives you, in fact, maybe I should, let me see if I got a, I think I got a sugar. Oh, I'm gonna, glucose. Here is glucose. Boo-ba-doo-ba-doo. Doo-ba-doo-ba-doo-ba-doo. Here's a glucose. And if we have our glucose, you know, I've talked about how, you know, oxygens, whenever you have oxygens, you've got those lone pair electrons that tend to make things more negative because you have a higher density of electron. Whenever you have a hydroxyl group, OH group, this O, which is actually connected to an H. And then this O also has this kind of more negative thing, and the hydrogen is going to be more positive. So when you have either oxygens like this, or we'll see later on the binding of serotonin and nitrogen in the ring, things are going to have things that like to make hydrogen bonds. Hydroxyls tend to like to make hydrogen bonds because they have this local charge imbalance, the oxygen versus the hydrogen. So glucose does not dissociate. They're covalent bonds, the thing stays intact, but it does dissolve. It does like to go into solution, dance with the other polar water molecules and dissolve. So it dissolves, but not dissociates. So glucose. You know, in terms of the actual lab itself, it's normally you would do it, but instead what you're going to do is just write down what you observe Tina and I doing. So in your lab notebook, I'm going to like give you a few moments in a second. You know, the next part is going to be, you know, part 2, where you're going to be working with the electrolytes. You know, and you want to kind of write in real time what you see happening. You know, one is kind of just looking at the electrolytes in the little beaker. What does it look like? You know, spoiler alert, they all look kind of like salt. They look kind of like little white crystals. Um, you know, then you're going to add a little water and see, do they dissolve? This is one of these things where normally I can do kind of a nice teaching moment, but we're not going to be able to do it because, you know, we're online. But very often people start adding a little bit of water to the salt or to the sugar, and they'll describe their results. They'll say, oh, let me describe this. It's, you know, there's a big oil slick, you know, when I dissolve salt or sugar in water, it seems to be creating an oil slick on top of the water. Like, just from your like intuition and your experience of spending a lifetime adding sugar and salt to things, do you suspect that adding, and just theoretically knowing what sugar and salt do when they go into water, would you expect them to make an oil slick on the water in the beaker?
No.
No.
No, not at all. So what do you think is probably happening when the students see an oil slick on the water?
Probably contaminated.
Exactly. Have you heard, Lauren? It's probably contaminated. You know, people have been putting oil in the beakers and then they didn't wash them well. So when you put the water in the beaker, you know, some of the oil that was still in there floated to the top. So this kind of brings in this idea. Maybe I can write it. You know, it's kind of what they call like a sanity check. You know, whenever you're doing stuff and you get some result that makes no sense, you know, kind of back up a second. And it could be in science, every once in a while, you get really surprising, non-intuitive results. So it doesn't mean it's wrong, but it means like if there's something that doesn't make sense, you want to kind of go back. The other thing, like for the jumping lab, it is not uncommon, particularly in people in our class who've just spent a lifetime learning about inches and feet. They'll measure somebody's height and they'll write down in their lab notebook, this is their height. Like, would this make any sense? How tall is 38 centimeters?
No, like a ruler, a little bigger.
It's a little bigger than a foot or so. So unless you are, having some like little elves as your test subject, that number can't make any sense. But then sometimes people just like, so I read off of the ruler, let's write it down. Again, it kind of goes back into this like, sanity check. Does this really make any sense? If you did some calculation with your numbers and you figured out that this canary weighs, 30 kilos, it can't, a canary can't weigh 60 pounds. That doesn't make any sense. So you made a mistake, which is fine. What's not fine is if you I guess I'll leave it as a 60 kilo canary, whatever. So as you're doing either measurements or calculations, kind of keep in mind, like you want to pay attention. Does my answer make sense? And sometimes it doesn't. I do calculations. I'll make some spreadsheet and I'll do all my formulas and I'll look at what's coming out and I'm like, holy crap, that something is wrong because numbers can't be that. So kind of keep that in mind. Kind of try to stay conscious. Don't just like record something because, or calculate something and write the number down if it doesn't make sense. So that's part of good lab technique is not just life technique, is kind of double checking yourself and kind of making sure that, because we all mess up, and you want to have these things in place to try to catch yourself when that happens, so it doesn't kind of propagate further out. Yeah, so in this, what we're going to do now, how many people have already kind of pasted in the procedure for the next part, like for part 2? How many people have not, I guess, I should say. All right, so what I would like people to do now is this is like 10:40. Let's come back in like 15 minutes and we'll start looking at the data for parts 2. But before that, I'd like you to like kind of, get out the scissors and paste again and put in the methods and procedure for part 2. So that's kind of there. And once that's there, we'll start gathering our data for that. So I'll be here if you have any questions, but I'll give you like 15 minutes right now to just assemble things. Let's see. Let's do this. So I'm going to share our Canvas. So again, all this stuff that we're going to be looking at now is on the website or on the Canvas site. So it's still available to you, even if, if you want to go back and look at it. And here we go. All right, so now we're going to do parts 2 and 3 of the chemistry review lab. And parts 2 and 3 are very similar to each other. It's just part 2 is dealing with electrolytes, and part 3 is going to do the same basic steps, but with carbohydrate in this case. And just to kind of give you a sense of kind of this mechanics of putting the lab book together. Here we have data down from the last part of the lab, probably leave a little space to kind of write up, but then we're gonna just have the instructions for part 2 here for electrolytes. I'm doing this quick. In reality, you would do this a little more neatly and thought out. But basically, I have the instructions for the lab right here. So, and you can use a glue stick, it's better than tape, but tape looks good too. And then I'm just going to follow. It's really important when you're doing the labs to follow it step by step. And we'll be reviewing, here are the broad overview of how the lab goes. But even if you think you know what you're doing, you still got to slow down and read step by step, because there's often subtleties. And also, often we're taking the data from many different groups, and we're averaging it all together. And you can't do that responsibly unless every group did the experiment in the exact same way. So it's really important, as much as you're gung-ho to just get data, to actually slow down, have at least like one person in your group who is the official kind of reader, gonna get more pedantic and So I'm gonna say part 1, use a spatula to add small equal amounts, a large pinch of the dry electrolytes, sodium chloride, potassium chloride, and calcium chloride to 3 separate 50 ml beakers. So these are the little 50 ml beakers here. They're very cute. And we're gonna put a pinch. A pinch is basically kind of what it sounds like. There's a picture in your lab book showing a little bit. It should be enough that you can actually see it, but not so much that it's not going to dissolve in a little bit of water if it wants to dissolve. We can come over here, and I've got each of these spatulas is labeled so I don't cross-contaminate between the different things. And I also probably want to make sure I mark what these are. use a pencil here, and I'm going to have some potassium chloride. I'm going to have a calcium chloride. That's another thing. In a lot of these labs, you're going to have very similar looking tubes or beakers, but with different stuff. And it's another place where you want to get kind of pedantic and be good about your record keeping and marking what's what so you don't get confused about things. But here, potassium chloride and a pinch, again, enough that you can see like the end of your fingernail, but not too much, because otherwise, it's, if you try to dissolve a whole cup of sugar in a teacup or something, it's ridiculous. So I'm going to put all of these in, and then our camera will come and look at this a little more closely. I've got my calcium chloride, get a little pinch, and I'm going to put that in that one. And I'm going to get my sodium chloride and put that in here. And we're going to now, all right. So I'm going to take these and put them like with a black background where I think it's going to be easier to see. And in each case, hopefully you'll be able to see there's this kind of these little white crystals at the base of each of them. Again, right here we have the sodium chloride, which is ordinary. All right, so again, this is, as we're doing this, you should write down in your lab notebook an actual observation. While you actually see it on the screen, you can describe what does it look like. And you can also use like, like the 2-month version of me helping describe it as well. But in your lab notebook, you actually do want to record, just what these look like before we do anything else, just as the little crystals sitting in there. So, table salt. This one is potassium chloride. This one is the calcium chloride. And this is where you're writing things in your lab book. When it says, oh, I just said it here. The appearance of the dry electrolytes. You actually want to record that in your lab notebook. Use your pen and write down the words that describe, granular, white, fluffy, whatever words. This is kind of a qualitative description here. So there's no right answer other than it should reflect what your observation is. And it needs to be something that you record while you're actually experiencing it. Memory is pretty imperfect. You don't want to go back and think, oh yeah, I think it was on a white powder later on. That's not what your lab notebook is about. That's not what science is about. Empirical data is what you observe. So you need to record while you are observing it. So I guess the basic, the important thing about taking data is that you record what you actually see. And sometimes it's not what you expect. And that's still okay. You write it down. It's true and honest record of your work, not like kind of what you would have seen in an ideal world or what you expect your teacher to you to write down. So now it says after I've actually, I would write down again like KCl. Right. So I should go back. Where is it? Hold on, I'm trying to get to where there was that view where you're actually looking down on them. There they are. So just, and I can say they all look pretty similar, kind of what I'm sure most of you have seen salt crystals before in your life. But it is important to record that. You know, because once we add the water, we can see, are they still there? Are they not there? When we do the same thing with oil, are they still there? Or are they not there? So to know whether or not they disappeared or not, you have to have a sense of what did it look like when they were there. And it can also, so all of these, sodium chloride, potassium chloride, and calcium chloride, what kind of bonds do they all got?
Ionic.
Chloride, chloride.
Yeah, they're all ionic bonds, so electrolytes. And actually, let me just briefly, if I take sodium chloride as a solid, and I put it into water, into an aqueous solution, what is the sodium chloride going to become?
It's going to
Sodium ion.
differentiate in sodium ions and chloride ions.
Yeah, exactly. It's going to be individual sodium ions, individual chloride ions. If I take potassium chloride as just a solid, as a crystal, what happens to that once it goes into the water?
Potassium ions and chloride ions.
This one's a little trickier. Calcium chloride. What happens to a calcium chloride when you put it into water?
You get calcium ion.
And calcium with 2 positive ions.
2 plus.
Right.
2 CLs.
And 1 Cl negative.
Now, so we have 2. So calcium chloride is also ionically bonded. So, but remember, calcium is 2 plus. Each Cl minus is 1 plus. So it takes 2 Cl minuses. So there's actually how many separate ions are free in the solution for every calcium chloride? Yes, we saw Morgan with is like triple P.
3, yeah.
Yeah, so, or is it like West Side or whatever? I don't know. So chloride, 2 chlorides and a calcium. So this 2 chloride, this means, you know, we could also write this as calcium plus chloride, right? So calcium chloride also creates ions, but it's going to be 3 ions. And again, this will be important when we look at the osmolarity, as we look at how many particles end up in the solution when you dissolve different kinds of compounds in the water. If we dissolve salt in the water, we're going to have 2 particles for every sodium chloride. If we dissolve calcium chloride, we're going to have 3 particles for every calcium chloride. So just kind of putting at, you know, all of these are ionically bonded electrolytes, but there are some important things that are interesting to look at, how they're different as well. All right. So the next step was to add water to these puppies. So now it says, after I've actually, you know, I would write down again, like, you know, KCl. In fact, I'll look at it. It's, you know, white fine grains, you know, etc. I'm not going to spend all the time doing it right now, but you would if you were here. Then I'm going to add water. It says add 5 mils of distilled water to each beaker, and then swirl the contents. To measure out the distilled water, I'm going to use a pipette. These pipettes are for much more accurate volume measurements. And you always want to use one of these little pipetter attachments here, which basically sucks up. You know, you don't want to ever use your mouth-to-mouth pipette because you might get something inside you don't want. These are a little confusing for people at first to understand. They come in different volumes. This one here is a 5 ml pipette, and it's designed to dispense volumes up to 5 milliliters. And the way it works is you would put it in the liquid you're trying to pipette, and the numbers actually kind of go upside down. They start at 4 at the bottom, and the 3 to 2 to 1, and 0 is up here. So the technique you would use is you actually suck up until you are at the 0. And that means now there is 5 mils in the pipette. And when I squirt water out, so this is just water, so this is actually safe. If I do that, this goes down to the 1. That means I have just dispensed 1 mil. I've now dispensed 2 mils, and I'm down to the 2. I've now dispensed 3 mils, I'm down to a 3. I got it all out, I have just dispensed all 5 mils. So, because I need 5 mils inside each of these, I can just pull up to the 0. I'm going to quickly, there's 5 mils there. Suck up again, up to the 0. 5 mils there. Zero. So here we've got all of our electrolytes. And again, electrolytes, you should know what it means to be an electrolyte. And what do potassium chloride, sodium chloride, calcium chloride all have in common? And I've just mixed them with water. And now, kind of take a look. And here we're trying to decide, record the appearance. Like, can you see it still? Did it dissolve? Did it disappear? I have to say, looking at these, they all pretty much look like they've dissolved. I don't really see the granules in there anymore. Right, so I mean, from there, sometimes it looks from the top like there's some white, but there's also white crap on the table underneath. So you have to trust me there that they were dissolved. And I should also just mention, I'll reiterate, there are gradations on the beakers to give you a ballpark of what the measurements are, but those are never accurate. You never trust those to be actual volumes beyond just kind of ballpark, so you know kind of where you're close to. If you want to actually measure out precise volumes, you need to use either pipettes or graduated cylinders, or volumetric flasks is another way to go. But the gradations on a beaker are never something you use to actually get a accurate measurement that you care about. If you care about it really being spot on, rather like, kind of close. And then the next part is going to be testing for electrical continuity, basically whether or not it can conduct electricity. And conducting electricity is basically, an electric current is moving charge. So for there to be an electric current, there has to be charge moving, in the wires, in the lighting in the room, it's electrons moving through the wires. In most biological systems, when you have electricity, the electric current is ions that are moving. So having ions, independent ions floating around in a solution typically is going to allow an electric current to flow. We can test that. I have this nifty little thing set up here. It's actually just a power supply. A little buzzer. This thing's a little buzzer. and wires that if I connect these two wires together, it completes the circuit, and you should be able to hear the buzzer. So wires are separate, no electrical current flowing from one wire to the other. I don't hear anything. I have the two copper wires touching. I hear it. So this is just a really simple way to see if something conducts electricity well. You know, the air right now is not conducting electricity very well because I don't hear a buzzer there's no electric current flowing through the air from wire to wire when they're separated. I can take one of these solutions, and again, these wires are not touching, but if I put the solution flooding, so both wires are underneath and submerged by the solution, I can see if I hear the buzzer going. And when you do this, you want to make sure that the wires have a lot of surface area in contact with the solution. If you just like barely dip their noses down, there's so little contact, you know, you're not really going to get a good answer. But here, I'm going to take these, I'm going to put them in. This right here is the calcium chloride. Right, so this is also stuff you're going to be, so hopefully you wrote down now whether it dissolved or not, which we saw they all dissolved. Now we're looking for electrical continuity, how well each of these solutions conducts electricity. Can the electric current flow from wire to wire through the solution, and which will be evidenced by, let's find out, with bated breath. Here we go, let's see what happens. All right, so on the air, I don't hear anything. In calcium chloride, I hear a buzz. I can rinse these little guys off with some distilled water, because I want to make sure I'm not kind of cross-contaminating stuff. And normally I would just have like a little beaker like that. So I'm cleaning these off. Now I'm going to go into my sodium chloride. And again, same thing. Air, there's no electrical current. I'm going to submerge them in the sodium chloride. I don't know if you can hear that. I definitely you can hear the little buzz go. And then we can look for the continuity. We put the wires, and they do not they don't make a current in the air, but if they are submerged under the potassium chloride, I can hear a buzz. So again, I'm kind of in parallel, so they're not touching. Want to make sure that there's a large surface area submerged, and sure enough. So that is that. The last part is it says pick any of those electrolytes. And all right, so hopefully you got that down. That's, pretty clear. It's pretty clear how these get their name electrolytes, right? Again, for the purposes of our class, it's going to be a critical feature of these ions is that they carry electric currents. That's how, again, all the electrical signals in your nerves and your muscular system is all dealing with ions. So the final part is we're going to now, instead of using a polar solvent, which is water, we're going to see what happens if we put an electrolyte in a non-polar solvent. In this case, it's going to be oil. So we're going to do the same kind of thing, look for dissolving, look for electrical continuity. What do people expect? What do you expect if you are going to put salt in oil? Is it going to dissolve?
No.
No, because
No,
remember that like dissolves like. Polar things don't usually dissolve in non-polar. You think it's going to conduct electricity?
No,
Why is that?
because they will not dissolve or dissociate in
Yeah, so
non-polar solution.
There's no dissociated ions in the solution. They're all still just holding on to each other in their crystals. So there's no free ions to conduct electricity. So let's see what happens. Now, instead of dissolving them in water, put them in oil. So let's do that. Okay, I'm going to take one of my little beakers. And it doesn't matter which electrolyte I pick. I'll pick salt because it's the one that I am, table salt, the one I'm most familiar with in my life. Take some. Again, take a pinch. And I add it into my little beaker. And I don't need to label this because it's only one electrolyte. So this is the salt. If you take a look at it, it looks like salt. And now I'm going to take the mineral oil, and I can take 5 mLs of mineral oil. In the same basic way, suck up to the 0. I now have 5 mLs in my tube, and then dispense. That's going to be 5 mLs of oil that are now in my beaker. And now I kind of mix it up. I don't know if you can tell, if you can see the crystals. I don't see the crystals dissolving. I just see a bunch of crystals lying in the oil there. I don't know if it's possible. Does that show up there? And then I can check continuity with the electrical continuity. And you can do this at home. If you've never done it, it's kind of cool. Just take a little bit of salt or sugar for that matter, and put some in water and put some in oil. Most people got oil at home, and really kind of prove to yourself that salt and sugar, these polar compounds, are not going to dissolve in oil, that they just stay intact as their own little crystals. So again, you can see it on here, but I highly encourage you, if it's not something you have like real world experience with, put some salt or sugar and some oil and notice that they don't dissolve. Then I take my wires, kind of holding them parallel so they're not touching, and I'm going to submerge them in this mineral oil salt solution and make sure they're fully submerged. And now I don't hear anything. Right? Air, no buzz. Oil and sodium chloride, no buzz. Again, you can double check, fail safe, make sure the system's actually working. If I touch the wires, I totally hear a buzz. So the system is working. It's just I did not have an electric current when I was testing my solution. And that's more data I'd be recording in my lab notebook. And a little later on, I'll be spending more time kind of interpreting what does it all mean. But the first step is while you're actually getting the data is just to keep an accurate record of your observations. All right, so that is part 2. That is the data. So at this point, hopefully you have kind of recorded the data from looking at what does the salts look like? What happens? Do they dissolve? Do they conduct electricity? What happens when you add the salt to the oil? So at this point, you can, if you can tape in the last part, which is part 3, the carbohydrate one. So you should put that in now. Again, take a few minutes and put that in your lab notebook. And then the discussion you can do after the data for part 3, because part 2 and part 3 have a discussion that is basically integrated with each other.
I have a question. So for the questions that are on these papers, like how do water and oil differ in their relationship, should we be writing this kind of with our observations as we go, or should we wait to answer those questions in the discussion?
That's more of a discussion thing.
Okay.
Yeah, no, the data and results is more kind of your empirical observations for the most part.
Would it be okay to write it under the observations just so we can write it as we go through the videos? Or should we rewrite it in the discussion as well, just so when you're reading it?
Yeah, even if you do write it, you'll have to rewrite it, because the discussion should be where I see you take all the concepts and put them together and tie in your observations to the theoretical concepts and all that.
Okay, thank you.
And I froze myself on, I look kind of dorky here. Hold on.
Is electrical continuity the ability to transmit charge or current?
Electric current. Electrical continuity means that an electric current can flow through it. So the classic term continuity tester is something that tells you whether or not this thing conducts electricity. All right, is there anybody not ready to continue? All right. So this next part is similar, except we're using glucose now, sugar. And here we go. Getting the data is just to keep an accurate record of your observations. All right, so now we're on part 3 of the chemistry review lab, which is basically the same thing as part 2, except instead of using an electrolyte, we're using a carbohydrate, in this case, glucose. So I'm gonna get my little 50 ml beaker, and I'm gonna put a pinch of glucose in there. And I go find the glucose. I find the appropriate labeled glucose spatula. And 1 pinch of glucose. There's my pinch of glucose, my beaker, put it in. And again, you can kind of take a look and see there's a little granulated glucose crystals in there, which again, you would record in your lab notebook. Then you're gonna, that was really fast, but hopefully you can see. And right, glucose is not sucrose. Most of the sugar like the sugar you're putting into your coffee or something, that's sucrose, which is a disaccharide, which we're going to talk about in more detail on Thursday, but it looks pretty similar. Pull up 5 mils of distilled water. I'm going to add the 5 mils of distilled water. Agitate, give it a chance to dissolve if it's going to dissolve. And you can take a look. And it's worth, especially if you still have your other beakers out. Like I have a beaker of sodium chloride here. They both look pretty much the same. What does dissolve mean? The crystals have gone into solution. But then we're going to look at another thing. So here we looked at, does it dissolve or not, which we did with the electrolytes. We can now look at the electrical continuity. Again, double checking. Okay. System is online. I'm going to hold my little wires parallel so they're not touching. And now I'm going to fully submerge them under this sugar water. And I don't hear anything. So that's telling me something about the electrical conduction of sugar water. And again, if you remember, if we were looking at the sodium chloride, and it looks physically exactly the same. But if I put the wires in there, it's a very different response. So like in your discussion, parts 2 and part 3, even though they're kind of separate parts, in your discussion, you should probably kind of integrate your observations for both the electrolytes and carbohydrates under the same conditions and how they are similar and how they're different and why that makes sense. The last part of the carbohydrate part is just looking at the optical difference of what. All right, so just before we go to that last part, so just that I'll reiterate what I just said. I'll say again, the discussion, which you'll write after, after we're done with all this, make sure you talk about how your observations, illustrate a lot of these basic ideas we've been talking about what does it mean to dissolve or not dissolve, or dissociate or not dissociate, and how that affects things. And so make sure you kind of pull that together. For this very last part, I realized I was hoping to get through what colloids were before this moment, but I need to take a second to talk about colloids. So I'm going to do a quick little detour here, so this last piece makes sense. So when you're mixing things together, usually there's some kind of solvent, which is the preponderance of stuff, and then some kind of solute, the stuff that you are kind of mixing into the solvent. In sugar water, sugar was the solute and water was the solvent to make sugar water. And sugar water and salt water and all of those are what we call true solutions. In a true solution, there's 2 qualities that make something a true solution. One is the solute never settles. So if you mix, assuming you're not over, putting too much, if you mix up some sugar water or salt water or anything else that's a true solution, and you just wait overnight, it's still going to be in solution. You're not going to have mix the sugar in there and come back tomorrow when all the sugars got pulled to the bottom. So a true solution, the solute never settles. 2 does not scatter light. What that means is it looks glassy. You know, it might even have a color. It might, but it looks clear. It looks kind of clear. You don't. It doesn't look milky or cloudy or anything. It looks transparent. So by not scatter light means it's kind of transparent. And again, examples, and that's because the particles, the solute particles are so small, they don't affect the passage of light. So that's true solutions. And again, most of the stuff we're going to be talking about, or a lot of the stuff we're talking about are true solutions. Again, the sugar water, the salt water. Another thing that's going to come up in just a few moments to end our lab are colloids. So colloids are when the solute particles tend to be large, like much larger than the ones that make a true solution. So there's still, the solute never settles. So things do stay in solution, but the big difference is that they do scatter light. Like if you look at them, they tend to look kind of milky. An example of this would be putting starch into solution. So the starch, we're going to talk about starch in more detail on Thursday, but starch is a big molecule, a polysaccharide, lots and lots of glucoses all bonded together in big chains. You can get it to go into solution. It's kind of more of a pain. You got to heat it up and mix it for a long time, but eventually it'll go into solution. Once it's there, it will not settle. But when you hold it up to a light and try to look through it, it's going to look different than like just sugar water. If you hold up sugar water and hold up a starch solution, they're going to look different, which you're going to see in a few moments in the video. Colloids also have all sorts of other interesting properties that we're not going to talk about so much in this class. They exhibit what are called gel to sol transitions, meaning gel, meaning they're more kind of gelatinous, sol, meaning they're more liquid. So sometimes just small changes, the whole thing will all of a sudden be more kind of solid and then more flowing. The cytosol in a cell actually can do stuff like this. So colloids are actually important, but for our class, we're not going to be looking at them as directly. The one other thing that's not a true solution or a colloid are suspensions, which I should mention those as well. Whereas solutions never settle, colloids, solute never settles. If the solute particles are so big that gravity eventually pulls them to the bottom, we now call it a suspension. I mean, if you've ever taken muddy water and then left a glass of muddy water on the table overnight, when you first look at the muddy water, it just looks like, oh, gross, muddy water. And then the next day, you have a layer of silt at the bottom and totally clear water on the top. That's a suspension. Your blood's a suspension. Where later on in the semester, we're going to pretend we're doing hematocrits instead of doing them. But basically your blood is a bunch of blood cells in your plasma. But with enough time, the blood cells fall to the bottom, or in it, when you're doing hematocrit, you put it in a centrifuge and speed up the process, and you just get the blood cells all packed on the bottom and the plasma still liquid on the top. That's an example of a suspension. Gravity can pull these solute particles down to the bottom. Clinically, it's important to know if like a medicine is a suspension or not. A lot of times you'll read the label on something and it will say it is a suspension, which means what do you need to do before you feed it to somebody? Yeah, exactly. See Morgan's got like all the hand moves. This is more like the shimmy or whatever. The shaking it up is going to be critical, or else you're going to like just give them the carrier fluid and you're not going to give them the good stuff that all settled into the bottom. So it's important to know if something's a suspension, because that means you got to shake it to re-suspend stuff before you actually use it. So basically I had to make this little detour here because when we compare, an example of a true solution is just dissolving glucose in water. An example of a colloid would be dissolving starch in water. And let's take a look what it looks like. And the starch was not dissolved great in this one, but it's still visible enough that it's worth looking at here. So we're going to go back. So in your lab notebook again, kind of just to orient, we are now doing the final step of part 3 of chemistry review lab, which is basically comparing a jar filled with glucose solution to a jar filled with starch solution and kind of noting their optical properties. And here we go. Sugar, water, glucose solution looks like compared to starch solution. In starch, which is just a whole bunch of glucoses bonded together in big long chains, but when they go, and it will go into solution, but becomes a colloid. And it's worth taking a look here. Now this is the sugar water, a true solution, which by definition is not going to scatter light, is going to basically be transparent. Whereas when you have a starch solution, I don't know if you can kind of notice it here, a colloid does scatter light, which basically means it's kind of cloudy or milky looking. And hopefully that, part of, this is a demonstration lab, trying to get you to just have a more personal sense of like, oh yeah, I see that glucose dissolves into a transparent solution, whereas the starch is dissolving as well, but it's creating a different kind of mixture, this colloid, which you can physically see as being different. So, look at that. So after me, they've got Will Ferrell and Jim Carrey. Yeah, so I'm inspired. John Cleese and, oh my God, Bob Ross. I have to say, he's helped me kind of feel a little less freaked out about the world imploding. So yeah, if anybody needs to like just forget that everything is scary, you can go watch a half hour of him painting and it'll help. Let me stop sharing. So, are there any questions about any of this?
Do you want us to combine the discussion for part 2 and part 3 then?
Yes, that because they are kind of just, telling a more integrated story.
Okay.
Doing the same kind of things. And it also is going to be a good opportunity to really think about the similarities and differences between covalent polar things that dissolve in water versus ionically bonded things that are also dissolving but doing so much more. So in terms of the expectations for this, the next step is going to be doing the kind of finishing up the write-up. And then there'll be the scanning using your little iPhone apps and scanning and making a PDF that then you can upload to the Canvas site. So in that, I forget when I made that due. Let me see when I made that due, actually. I don't even remember now. Let's see. Doing the chemistry lab.
Friday, the 4th.
Friday the 4th. Okay, so that's so the end of the week. And it's partly to just try to keep you moving on it so you don't like leave everything for the last minute. I mentioned at the beginning of the semester, I'll mention again, if you don't get something in on the official due date, please still do it and turn it in. You'll still get most of the credit for it. So don't think like, oh, I missed a due date, I might as well just forget about it and not turn it in. That's gonna hurt you bad. So the other thing we're gonna do today, what time is it? So we have like 45 minutes here. I'm gonna spend maybe we'll take another maybe a 10-minute break, but then I wanna talk just a little bit about the idea of orders of magnitude, the idea of accuracy and precision, and then talk about the preparing for your jumping lab. And there is going to be a little pre-lab. It's only going to take you like 10 minutes, but there is something that you need to think about and physically do before we actually do our lab on Thursday for the next lab. But let's take it's 11:41. Let's just round it up. How about at 5 minutes? We could even do, let's do 5 minutes to noon. So 11:55, we will all meet back here and we will tie this all up. So here we go.
Professor, do you need a break, or can I ask you a few questions? I can also do it after class.
No, this is good.
Okay, I have some questions. I think I'll have to do some studying on my own, but just, you mentioned a few times like the phosphate, glucose, and then the creatine. And if we could go over that in a basic sense so that I can have an understanding of what you're talking about.
Okay, so that was an example I was using of an exchange or displacement reaction, and also it's a nice example of another kinase, actually.
Right.
So for instance, so remember ATP breaks apart to become ADP plus phosphate.
And I think that's something like I need to go back and understand that better too. Do you think you could do like a little tiny overview of that, and then I will on my own time?
Yeah, so the basic idea, the A, and I'm going to keep it in more of a cartoon way rather than, it doesn't matter the detailed atomic structure of this.
Okay.
The A is basically the nucleotide base, the adenosine, that you have in the nucleic acids. And then it has 3 phosphate groups attached. These are like a phosphorus and 3 oxygens each, not 3, 4 oxygens each. So you have the nucleotide base, then with 3 phosphate groups attached. And there's a lot of energy that is stored in these bonds between the phosphates. So if we have a reaction where we take this and we break off one of the phosphates, so we end up with this thing, which we now call ADP, adenosine diphosphate. And this thing here was ATP, adenosine triphosphate. And the phosphate that is been broken off, since it's no longer connected to an organic molecule, this P sub I means inorganic phosphate. So this is going to be one of the main ways of supplying energy to processes in the body, whether you're doing muscle contraction or pumping molecules against their concentration gradient or all sorts of things. So we're going to see this a lot. ATP is broken apart into ADP plus phosphate plus releasing energy. So again, you can think of this as the thing that has the potential energy, and this is when you release that energy to do something interesting with it.
Okay. That makes sense to me. And then just so I understand, when you said like the adenosine is attached to the triphosphate, and then is the phosphate also attached to different things as well? Or is it just phosphate? Is it like, what is it? Because you said phosphate groups.
Yeah, each of these P's represents a phosphate group.
Okay.
Yeah, I could, here, hold on. What I need to get into, here we go. I'm gonna look at I'm just gonna go ATP. Right, so do you see that guy?
Yeah.
So that is the actual molecular kind of formula for this thing. And whoop, I need to draw. Here we go. Wait, why isn't this working? Wait a second. For some reason, my annotation isn't working properly. So let me try this one. Let me try this one more time. I'm going to share. Oh, now I think it's going to work. Okay. Phosphate, phosphate, phosphate. This is adenosine.
Okay.
This is A, P, P, P.
Okay. And so to repeat it back to you, it starts off as the adenosine triphosphate because there's the three phosphate groups, and that is the stored like potential energy that can be released, right?
Right.
And then when one of the phosphate groups breaks off, it becomes inorganic because it's no longer attached to an organic. So, and then is it the breaking off that is the release of the energy? Oh, yeah, and then the other ones become ADP because it's diphosphate, 2-phosphate groups.
So it's the rearranging the chemical bonds releases the energy.
Okay.
During that breaking apart, the ADP, the A from with these two, breaking this this bond right here is what releases the energy.
Okay.
So
Got it, I think. Okay. Thank you for going over that.
sure.
Appreciate it. Is that worth, I mean, does it need to be a more complex understanding of that for this class, or is this what I just need to know? And
So in this, I didn't mention the words, but this would be an example of what we call an exergonic reaction. Let me go back. So there are some reactions where you have to feed them energy to make them work, and other reactions that give off energy when they run. So ATP breaking apart into ADP plus phosphate will release energy, which can be captured to do stuff.
okay. Just writing down. Okay. Got it.
And then I can add in the thing I mentioned about the kinase.
Yeah.
So let's say you've done this, like this happens during muscle contraction. You're using the ATP in your muscle tissue, breaking it apart, using that energy to cock the little myosin heads that are going to generate the forces.
Right.
And especially in your fast twitch muscles where you want to have ATP ready to go right away again, you've got this system of creatine phosphate lying around. And so if you have ADP, which is again the A with the 2 P's, and creatine phosphate, which is C with 1 P, this is creatine phosphate. You know, this is just another, you know, this is a nitrogen containing thing. You know, don't worry about the detail. The creatine is just another thing that, just like adenosine had the 2 P's on it, here's the creatine that has 1 P on it. Then you're going to go along with some enzyme. It's going to be this creatine kinase, actually. Kinase because it's transferring a phosphate group. And basically this phosphate is going to get slapped back onto this. So we're going to end up with ATP because now we have just moved that phosphate group. And now the creatine is kind of standing alone because you transfer that phosphate group from the creatine phosphate onto the ATP. So now in one step, you have regenerated the ATP, so it's ready to go to help with generating more muscle force.
Okay.
Okay, we're going to look at this in way more detail when we do muscle physiology. It's one of the ways, when we look at ways that muscle generates ATP, this is the fastest way, but you have very limited amounts of this lying around. This only lasts for about less than 10 seconds. Like if you're sprinting, you could make ATP really fast this way, but it's going to run out pretty quick. But this is just an example of an enzyme facilitating a reaction. It's the example of a displacement reaction where you're moving something from one thing to another. And it's also an example of phosphorylation. Which is going to be a recurrent theme in the class. We're going to turn channels or enzymes on and off by phosphorylating them. We're going to be doing that a lot, adding phosphate groups, taking phosphate groups off of things to control activities in the cell.
Okay. Thank you. I understand I understand the broader picture, just not necessarily, I think, like the intricacies of how the phosphate would then add back on to the ADP, or like, you know, actually how that is working. But I understand like the concept of what you're talking about.
For this class, you don't need to know the intricacies. And I probably couldn't, even if you pushed me, like how are the orbitals lining up? Yeah,
Okay.
that's not what I care about.
Got
I care about it more on an energetic level.
Okay, then I can do that. That makes sense. And then thank you for going over that. And then another question is, in terms of workload for this class, do you think it's the same as anatomy in terms of how much time studying outside of class, et cetera? Do you think they're comparable, or how would you?
I think they're comparable.
Okay.
Yeah.
Got it.
They're different. Anatomy has a lot more rote memorization.
Yeah.
This class has a lot more conceptual things that you have to kind of bang your head against the wall to figure. It might not be as much stuff, but it's very different kind of stuff.
Right. And is chemistry for the whole time such a essential part of it through the whole class?
Yeah, it will be.
Okay.
Yeah, because at the very end, even we're just doing like reproductive systems. The last system, it's the core is looking at molecular interactions of hormones and receptors and things like that.
Okay.
That's gonna, and how do steroid hormones get into the bloodstream? They need special carriers because they're lipids and they don't dissolve because they're nonpolar. So if you were confused about nonpolar things not dissolving in water, that would be like, why? Or,
I get all that. That stuff makes sense to me.
cool.
Some of these other things. Thank you for going with that. Appreciate it.
All right, so we are at 11:56. So we are gonna do a couple of things here and then we'll call it a day. One thing I want to talk about is the jumping lab, which is coming up on Thursday, rescheduled for Thursday, this coming Thursday. This is going to be the first of your labs where you actually have a hypothesis-driven lab. The chemistry lab is a demonstration lab. There's no, the objective, like demonstrate stuff. But the jumping lab, there is an actual question, an independent variable, a dependent variable, which you need to clearly state in your lab notebook so we know exactly what's the whole point of doing this. Then you need to come up with a hypothesis. Again, what relationship do you predict between these variables and why? It has to be both the what you expect and why that makes sense, the educated guess. It can't just be a random guess. It has to be a guess that has been derived from some thought process. And then the last piece are sources of variability. You have to think about in advance what kind of things might affect our results that aren't necessarily the things we're looking at, but actually might be important to take into consideration so they don't destroy our results. So I'm going to go back and share. I'm going to go, here it is. I'm going to go share our Canvas screen again. So I've been adding some stuff in here. Ooh, too far. Here we are. Jumping lab, lab number 2. One is there is just a copy of the lab manual in here. So I want to view it. Why doesn't it open to view? Oh, there. There's file preview. Come on, puppy. So jumping lab. You can read about it here. So basically, you are going to gather data for people. You're going to measure their weight. You're going to measure their height with shoes off and the calf circumference. You are then going to have them do this vertical jump, and this, they call it this, I'm calling it the standing calf jump. And then we're gonna like see if there's relationships. So for the first part of this getting ready, you need to be able to very clearly write out what is the objective and write it in your actual lab notebook. So let me stop this preview. Again, so if you don't have the lab manual, if I know if people were having some trouble, you have the actual lab manual pages there for you. Let me go. So pre-lab. So I would say first you're gonna like kind of paste in procedure. So this is gonna be the first thing you're gonna do is in your lab notebook, this is gonna be like after your discussion for the chemistry lab. You know, and if you haven't totally finished the discussion when you're decide to do this, you know, give yourself a little bit of space. So paste in the procedure for this jumping lab, and then this pre-lab is going to be objective. And again, we've talked about this a bunch. Objective is just going to be some very specific statement about, this is a question. Let me try to do this. So this is always a question. And it's, you know, basically what is relationship between the independent variable and dependent variables. So what are the independent and dependent variables for this jumping lab? So if we are measuring, we're measuring people's height, their weight, their calf circumference, and then seeing how high they can vertically jump. What
So
would
the independent variables would be the height, weight, and calf circumference.
right? So these are the things that we're going to measure. We're going to look at different values of these, and then we're going to see, do they have some relationship, some predictable relationship to our dependent variable? Again, which is what? What's our dependent variable?
How high you jump.
Yeah, it's the height of the vertical jump. So what could be really a real simple statement for the objective for this lab? You're just going to put it into words.
This lab.
So
how high you can jump will be indirectly proportional to the subject's height, weight, and the calf circumference.
So you've got the right variables, but you're kind of jumping the gun in that you're kind of giving me an answer already rather than just stating it as a question. So the first part is the question. What is the question we're going to try to answer? The next part is going to be the hypothesis, what you expect might be what you find, but we have to start more in the realm of question. So something that's a question.
Does the question could be, does height, weight, and cost are related of how high an individual can jump?
Yeah, exactly. So something as simple as that, like how is a subject's height, weight, and or calf circumference related to how high they can vertically jump? Something like that is all you need. Or what is the relationship between the height, weight, and calf circumference and the height of their vertical jump? Or, how is height of vertical jump correlated with a subject's height, weight, and calf circumference? Or there's not one correct statement, but it should just be a very simple, concise question that relates how are these independent variables related to our dependent variable. And again, it's a question. You don't know what the answer is yet. And again, like I mentioned last week, the first part of any experiment is having a clear idea of what exactly are you trying to get at with this experiment. So once we know specifically their height, their weight, and their calf circumference related to the height of their jump, then we know how to do the experiment. We're going to have to measure their heights and their weights and their calf circumference and the height of their jumps. So that is part one. This pre-lab has 3 parts. Part one is this objective, which again is just like one sentence, but it's a well-crafted sentence that gets at the heart of what this whole experiment is about. And this should be written in your lab manual before Thursday morning. So before we actually do the experiment, I want you to just very clear, I want to be convinced that you know exactly what we're up to and why we're doing it. Part two is going to be the hypothesis and the educated guess. So maybe Jamba, what do you think might be, what do you think the relationship might be here? I can't hear you actually.
If you're heavier on the weight, then you might not be able to jump as high versus the person who has a lesser weight can jump higher.
All right, so here he has a hypothesis that weight, someone's weight is going to be inversely related to their jump height. And remember, it's always got to have the what and the why. So he's saying in his thing that heavier people will have lower jumps. And then why do you think that would be the case?
It's more of a load.
Yeah, I mean, we're definitely one of the things, in order to jump, you're fighting gravity. You're having to push a mass up against gravity. So if you want to have your complete hypothesis, you'd have to say, I think heavier people will not jump as high because, their muscles are going to have to do more work against gravity, so something like that.
Is there the variable, the energy level of the person?
That's going to be more of when we talk about sources of variability, that's going to be an important one, but that's not going to be one of the ones that we are actually measuring. So your hypothesis must, address the independent and dependent variables. So, energy might, somebody who didn't sleep last night very well might have a horrible jump. But that shouldn't be part of your hypothesis because, that's not one of the specific variables we're looking at. Unless you say, like, I don't think any of these variables are going to be correlated with the jump height because there are so many other confounding things such as energy level, because we don't know how much they slept or whatever, right? So, but in general, you need to make sure your hypothesis addresses this relationship. Again, the question is, what is the relationship between the independent and dependent variable? Your hypothesis is, what is the relationship you predict?
Do we need to speak to each of the independent variables, or can we just say, I think heavier people are going to have a lower jump?
You can just speak to one. If there's one particular one that you think is particularly intriguing, you can just use that.
Okay.
Eventually, I would say for our more complete analysis that we're going to be doing next week, we're going to look at all of them. Once we actually get all the data and then we have the tools to graph things in Excel, then it will be required to actually look at everything and make sure that you're not missing any relationships. But for this first step, I would like you to just come up with the hypothesis that makes sense to you.
Okay. Thank you.
Sure. What's another possible hypothesis?
The greater someone's calf circumference, the higher they will jump, because the more muscle capacity they have to lift off the ground.
Yeah, so that's right. So we're not actually measuring muscles. We're actually measuring just calf circumference. But so in that Y, you probably want to be a little more explicit, like the larger a calf circumference, the more likely it is to have hefty calf muscles to generate the force to jump off the ground. But the danger is, if you do a cross section of someone's calf, it could be just lots of muscle, or it could be a little bit of muscle and lots of fat. Right? So calf circumference is not a direct measure of muscle, but we put it in there because it's likely. It could be. And this actually brings up the other thing that I have in the lab manual about BMI. So let's mention BMI briefly. And this is, what is it? It's, oh my God, height, no, kilo, it's the weight over height squared, is it? Yeah. So this is like a ballpark, kind of comparing how much does somebody weigh to how high are they, with the idea that if you have two people that are the same height, if somebody has a BMI, like when BMI is between 18 to 25, this is what they call kind of the healthy BMI. It means that your height to weight ratio seems to be in this healthy range. When BMI is 25 to 30, your amount of weight for a certain height is getting into unhealthy. When you are greater than 30, this is starting to consider obese. If you're less than 18, this is also unhealthy. You're too scrawny. You're kind of getting anorexic or something. But so this is, again, a ballpark, though. It's important to remember that it's not an actual measure of your fat to lean tissue. It's just kind of ballpark. There are ways to actually get your actual fat to lean body mass using underwater weighing or calipers or things like that. but this is kind of a quick and dirty way to, like, in general, see if somebody has a good ratio. So if you were actually, and we're going to calculate BMI for each subject. So back to Sari's thing, like, your hypotheses might bring in, like, I think calf is going to be related to, positively correlated with jump for the people with healthy BMI, because in those people, calf circumference is more likely actually related to their muscle mass in their calf. So you can do something like that if you want to be a little more kind of nuanced about it all. BMI, again, doesn't tell you for real. Like, one of the things I wrote in there was Arnie Schwarzenegger had a BMI over 30 during the peak of his competition, because he was just pure muscle. There was not an inch of fat on him. So he was really heavy because he was so heavy, so bulked out for his height. So just in guys in general tend to have a larger BMI than women due to muscle. Like a guy, in general, testosterone is anabolic. Men and women doing the exact same things, the guy's going to end up with heavier muscles. Women end up having more body fat and subcutaneous fat just because of estrogen. So in general, it's more likely, like in guys, that it's possible that they have a higher BMI, but it's actually due to muscle mass because they're ripped or whatever. So you can't trust just because BMI, you look in a chart and it says healthy or unhealthy. You need to be careful about interpreting it. It's not actually giving you genuine fat to lean body, but it's a useful enough tool that we use it, obviously. So you can have other hypotheses. There's no right or wrong hypothesis. The idea of doing an experiment is we don't know what the answer is. So come up with something that makes sense to you, but just make sure that you have the reason as well. You always gotta have the why. You can't just say larger calf, higher jump. You have to say larger calf, higher jump, because I think the larger calf is gonna mean like more calf muscle to generate force to jump. So make sure you have the why. And then finally, so this is 2. Number 1 was the objective. Part 1 of this pre-lab is the objective. Part 2 of the pre-lab is this hypothesis, again, which is just another sentence or 2. But we're not talking, you got to think it through. But this is once you know what you're going to write, it's just a sentence or 2. And then the final part, part 3, sources of variability. Again, for this jumping lab, what would be sources of variability? Is there any of the variables that are not our independent or dependent variable that might affect things?
Injury.
Level of energy.
Injuries, level of energy.
Genetics, training.
Genetics, training. These are all actually really important. Some of them we are going to be able to control for. Injuries we can deal with. We can make sure that any subject that has injuries that are going to prevent them from doing the jump they're excluded from the subject pool. Training, we're going to do a little bit here to try to make sure everybody knows how to do the basic jump. We're going to do some practicing to make sure they've got the moves down before they actually start recording data. Other things we can't control for. Genetics, level of energy. We could insist everybody has a certain amount of sleep or drinks a little Red Bull before they do whatever. But there's a limit to how much you can try to control everything. So you list them all out. You try to control the ones you can. You kind of stay mindful of the ones you can't as you're analyzing your data later to make sure that they're not messing you up. But this can just be a list. You know, this is a start, but there's lots more that can be on here. So just kind of sit and brainstorm and think about the things that might be affecting somebody's ability to jump vertically that are not those body measurements we talked about. And just list them here. And before we start the experiment, we can go down there and again, like try to make sure we take care of any of the ones we can so they don't mess up our results. So any comments or questions about that?
I have just one question. Approximately how many sentences do you want our discussion to be?
For what? For the chemistry?
Just in general, like an overview, or does it vary from experiment to experiment? Or is there like about like a paragraph, you know what I'm saying? Like a few sentences.
It varies radically. Like, you know, it could be like for something, a smaller little thing, it might be a half a page. For another thing, it might be many pages.
Okay.
Yeah, it's.
Professor, one question, me.
Uh-huh.
I don't know if you said it before, but which app for scanning the pages, because we need to submit this observations.
Okay, so so here I'm going to share screen again.
Thank you.
All right, so if you go here into lab orientation, lab 0 orientation, scanning using phone apps, I found that this Adobe scan, which you download on your phone, that was the one that I found worked the best for me. There are other ones. There's Genius Scan, which another one of the physics teachers likes to use. I didn't like it as well. But I would say you can start with the Adobe scan as a starting point. But if you want, feel free to use other apps as well. There's more than one app that works well for that. And what I would like you to do is submit PDF of Jumping Lab pre-lab. So this is here under Jumping Lab. Again, this is just going to be a page, right? This is just your objective, your hypothesis, and sources of variability. You know, and I just want you to send that to me so I can look at it before we start on Thursday morning and go look at each of you guys and give you feedback before we start to make sure everybody's on the right page or on the same page or is kind of, so this should not be a stressful thing. This should literally be a sentence for an objective, a sentence or two for your hypothesis, and a list of some sources of variability but it just means that you really have taken the time to make sure you know what you're doing as we start the Jumping Lab on Thursday.
And you want us to put the procedures before the hypothesis and the objective and everything?
Yeah, you should have those in your lab book, although in terms of this little PDF that you're sending me,
Don't need to.
it doesn't matter if I see those or not.
Okay.
What I care about is that I see your objective and your hypothesis. So